Okay, so let's dive into these notes we have. They're from a consultation, right? With an AI cooperative.
That's right. And it's focused on microscopy, some really tricky imaging problems researchers face.
Especially with complex biological stuff. So our mission here is to kind of pull out the main challenges.
Yeah, the core technical hurdles and also the clever solutions being proposed, AI, other tools.
It's amazing how even with fancy microscopes, you're still up against basic physics.
Absolutely. And just the nature of tissue itself.
Yeah,
The notes show how different methods hit different walls.
like fluorescence and confocal. You mentioned photobleaching.
Right, the imaging light itself damages the sample over time, bleaches it out.
So your signal gets weaker and weaker, makes long-term imaging really hard.
Exactly. It's like taking a photo where the flash burns the subject a little each time.
Okay. And then there's lightsheet microscopy. It's fast, generates loads of data.
Loads of data, yeah. Great for 3D. But, like the notes say, often you get uneven lighting.
Uh-huh. Dark corners.
Yeah.
Vignetting.
Yeah, vignetting, inconsistent brightness across the field. It's not just ugly, it messes up measurements.
Right, if you want to quantify things accurately.
Makes it much harder.
And what about the 3D aspect itself? Getting good detail deep into the sample.
That's a classic problem. The z-axis resolution, the depth information, it's just,
Yeah.
well, it's less sharp than the XY plane,
Inherently fuzzier.
pretty much.
Yeah.
And with thicker samples, it gets worse. Light scatters, things blur.
You get overlapping signals from different layers.
Exactly. Which kills your resolution even more. Seeing fine details deep inside. Tough.
So, okay, faced with all this, what are the actual technical puzzles they're trying to solve?
Well, one really interesting one is using expansion microscopy.
Expansion, you mean making the sample bigger?
Physically bigger, yeah. You infuse it with a swellable gel, then expand it uniformly.
Huh. So tiny structures become large enough to see with a standard microscope.
That's the idea. It's a neat trick solve an optical limit with chemistry and mechanics. Could help with resolution, maybe even the bleaching.
That is clever. And what about improving those blurry z-axis images, especially in brightfield?
Right, brightfield is common, but often struggles with depth. They're exploring computational fixes.
Like deconvolution? Or maybe training an AI?
Exactly. Could AI learn the blur pattern? Maybe if you train it using sharper data you already have.
Like from a confocal or lightsheet scan of a similar sample?
Precisely. Use the high-res data as a sort of ground truth to teach the AI how to de-blur the brightfield z-stacks.
Makes sense. And AI isn't just for fixing images, right? They're using it for analysis, too.
Oh yeah. Basic stuff, but time-consuming. Counting cells, outlining structures, segmentation.
Automating the grunt work.
Basically, it frees up researchers from clicking pixels for hours, lets them focus on the biology.
Okay, and this part was unique polarized light cameras.
Yeah, that's a fascinating angle, using cameras that are sensitive not just to brightness, but to the polarization of light.
And they hope this reveals something about biological behavior, squid specifically.
Right. Squid communication and vision heavily involve polarized light. So these cameras might let researchers observe or quantify behaviors invisible otherwise. Adds objectivity.
So lots of problems, lots of potential tools. What were the actual suggestions from the consultation?
It was definitely a mix and match approach. For plant cell fluorescence, dealing with bleaching and Z-blur, expansion microscopy was suggested.
And for the AI side, specific models.
Yeah, they mentioned noise-2-void N2V for denoising and segmentation. It's good because it can learn to denoise even without perfect, clean examples.
Useful. And for that brightfield z-axis.
Using something like deconvolution AI, trained on existing confocal or lightsheet data, the approach we just talked about.
Got it. And all this data, especially from lightsheet, it's huge, right?
Massive. So you need serious computing power, high-performance computing, cloud resources.
They mentioned grid computing, too.
Yeah, as an option to distribute the processing load, you just need a lot of processing muscle.
And for the polarization studies.
Adopting professional-grade polarization cameras. They mentioned Sony sensors specifically, to capture those signals reliably for the behavior research.
Okay. And there was one more broad suggestion. Something about a platform.
Right, establishing a more general image processing platform, a kind of standardized toolkit.
So different teams could use it for quantitative analysis.
Exactly. Ensure consistency, make comparisons easier across different imaging types and projects, standardize the analysis pipeline.
So this whole deep dive really shows, tackling these big biological imaging problems isn't just one thing.
No, it's bringing together optics, hardware, clever chemistry, like expansion microscopy, powerful AI, and just raw computing power.
Really diverse tools for complex challenges, which makes you think.
Yeah.
Well, it raises a question for you listening. When you run into a technical problem that seems impossible in your own field, what completely different area, what unrelated technology might actually hold the key?